So, our do now question again today was, what are different ways we try to kill bacteria or protect ourselves from bacteria? So, showering, washing our hands, brushing our teeth, cleaning anything we touch, using antibiotics, using heat, right? So, when we're cooking, this is part of why we always have to cook things to a certain temperature to make sure we kill off bacteria. Washing our hands, disinfecting surfaces, using bleach, wearing gloves. Avoid close contact if you know someone has a bacterial infection, get vaccination. So for really large, what used to also be epidemic level bacterial infections, we can get vaccinations, right, that help prevent those. Right, what do we clean our hands and surfaces with? So a lot of us mentioned, right, cleaning and sanitizing, but what do we actually use to clean and sanitize surfaces or our hands? Okay, what's in Clorox? Okay, so one is soap, right? Soap, bleach. Okay, good. What else? Do you use bleach every time you sanitize your hands or soap? Hand sanitizer, okay? And the main ingredient in hand sanitizer that kills bacteria is what? Does anyone know? What is the main ingredient? There we go, alcohol, good. So we are gonna build up to a point where we're gonna brainstorm the next segment from our bacteria. And we're going to think about how we could run tests on how effective these different methods are at actually killing bacteria, okay? Right, quick reminder that I do type everything in your plans here. You can access this from the home page in Canvas. So if you're in my Canvas course and you go to home, then biology daily plans, you would open up the same slideshow I show you in class. And it usually takes me about a day and a half or so to upload the previous class period's video. But if you look here, any of the dates that have this blue underlined link are actually the recordings of my teaching. So you just click on it, go to the video, and they are the recordings of me going over everything from class, okay? So if you are someone in here that has been absent a day, or frankly, maybe you're really behind, those are ways where in your own time outside of class, you can go back and watch those videos, catch up on the assignments, right? Because just like every class, I go over all the assignments. So you can rewatch the video. And unlike real time, you can pause it. So if you feel like I'm going too fast for you, that is always a way that you can kind of help get through it, okay? So don't forget about that as a resource. I'm going to go back in. So we did the 3.40 do now. We're now going to take a look at our results from our bacteria swabbing. Sorry. So about almost maybe 2 weeks ago, if you recall, we were talking about give me 1 second. Okay, so apologize by that. Sometimes staff members email me needing help with Canvas stuff. So our first investigation, we were just trying to see which areas of the school had higher or lower concentrations of bacteria. You guys had completed a survey to tell me which areas of the school you wanted me to swab. And I have the results right here. So let's take a second and look at these. Each of these is a Petri dish. You remember us talking about that. And inside is a clear, colorless gel, almost like a firm jello called agar. And you remember that that was the medium that has all the food and water how bacteria need to grow. So then I cover them in masking tape and I put them in Ziploc bags. Some of them are out of bags because it was easier to see what was inside the dish outside of the bag. And here is where we got the majority of our votes. So the majority of you wanted me to swab the cafeteria, a computer lab keyboard, the bottom of my shoe, water fountain. So I specifically swabbed the spout the water comes out of that you would drink from. The front door, light switch in the front office, behind the toilet, and the pool. So these are the results we have. Does anything here surprise anyone? For good or bad? Are any of these results, as we're looking at it, kind of shocking to some of you? They were to me. Behind the toilet, good, right? Look how pristine that Petri dish is. I could not believe that. I mean, I know that they have been very diligent about cleaning, but Khalil, I promise you, it's not fake. Because if it was fake, why would I try to make these look so bad? I promise you, it's not. Right. It's not. Well, they clean it. It actually was the staff bathroom. So I didn't use student bathrooms because we don't have that many students. And we don't have that many students that are regularly using the bathroom. So I used the staff bathroom, which gets used all day, every day. All right, front door. Pristine. You know, I have to say I was even impressed by the light switch. I know we have some growth in there, but you have to think the light switch is constantly touched all day, every day by secretaries, principals, custodians going in and out of the front office. So I was still impressed by that. A light switch I could see being something that maybe gets missed. Mikhail. Miss Aguilar said the same thing. So we both swabbed like one of the booths you can sit in. And we had the same thought, like there is literally going to be nothing here because no one is using it. It's all kind of closed off. And it was one of the worst, if not the worst, concerning. So I can only imagine what the cafeteria would be like had you all been here. Pool, I'm not shocked by the pool part because, you know, in all pools, they put so much chlorine in them, and especially any type of public pool. Chlorine, right, is the compound basis of bleach. And so its whole purpose in pools is to sanitize them. So I would be shocked if there were bacteria in there. Computer lab keyboard. So keyboards are huge harbors for bacteria, okay? And if I hold up my keyboard, it's not so much the surface of the keys, it's the grooves in between. But when you're typing, your finger is inherently grazing across all of that. And I remember one year, maybe about 3-4 years ago, we swabbed a teacher's keyboard. And this teacher was very adamant of like, you're not going to grow anything. I Clorox wipe my entire desk, keyboard, and computer every single day. And her keyboard was funky. Bottom of my shoe, I'm not surprised by that. I expect anything that's constantly rubbing against all different types of floors and the ground outside to be dirty, to be frank. Water fountain. What do we think about that? What do we think about the water fountain? Yeah, Michael. So the water fountain is my favorite story to tell. 2 years ago, we swabbed a water fountain. And again, what we swab is the hole or spout where the water actually comes out of. Because, yeah, we could swab the basin, but you aren't drinking water out of the basin. And we can swab the push handles, but unless you're like pushing it for water and then licking your hand, it's like, is that really that relevant? So we always swab the spout the water comes out. And 2 years ago, one of my classes, all of a sudden in class, they were looking at their dish and they were like, oh my God, Miss Roseman, I can see the bacteria moving around. And I was on the other side of the room like, that's not possible. What? I come over and in fact, inside their dish, maggots were growing, which means on the spout of the water fountain were fly eggs. Water fountains are one of the dirtiest places because people, they're fully open and exposed. People are constantly coughing and sneezing by them. In a high school, kids are always spitting in them. And all of that just collects in that area. So pretty gross. All right. So now it says your job is to click on the reply button below and post what your results from this experiment show us. Be detailed and thorough. So I want you to talk about just first in general, what do we observe in all of these plates? So you want to talk about all 8 plates. Then what does this tell us? Like which areas of the building have more traffic, which areas must be having less, or which areas are they doing a better job cleaning, things like that. Okay, so this is probably going to be about a paragraph long of writing. All right, so I'm going to give you, let's start with about 15 minutes. Okay, because it's not the only thing we're doing today. Again, you want to make sure you are detailed and thorough here. So you want to very briefly comment on the quantity of bacteria in each dish. And then you want to talk about, so what does this tell us about these areas of the school? And don't just put something like, the school's nasty, cafeteria is nasty, okay? I don't know what nasty means in scientific real life terms, okay? So click the reply button and start typing out your response here on our results. If you have questions on anything, make sure you drop it in the chat. Okay, in about 15 minutes, we want to be done with it so we can move on to the next part. So again, right now we should all be in Canvas. We're on 3.41. And we are typing out what we physically observe in all 8 dishes, and then discussing what this tells us about these locations in the school. Say about 6 more minutes. And we are in 3.41. We are posting a detailed response to what we see as our results from our first investigation. What do you observe in all 8 of the plates individually? And then what does this tell us about those areas of the school? All right, so we're gonna move on. You should have by now posted your reply to our results. And we're gonna update our evidence tracker here. Okay, so remember our evidence tracker is a place where we're kind of keeping just all of our different information that we figure out throughout this unit. We left off talking about the different types of bacteria, the parts of bacteria, types of antibiotics. Okay, so today we can kind of add to it what we discovered from the results of investigation part 1. So in general, if we had to kind of summarize it, why did we expect initially some areas of the school to have more or less bacteria? Why did we initially think some areas of the school would have more or less bacteria? Right, because of how often the object is used, how many people touch it, the amount of traffic in that area. So we initially predicted, right, that areas that had a lot of contact or traffic would have the most bacteria. Areas with very little contact would have the least amount of bacteria. Okay, but in fact, right, our results did not perfectly match up. So we saw that the shoe bottom, keyboard, water fountain, and cafeteria seat had the highest numbers of bacteria. Oh my God, does anyone else's nose like constantly get itchy in their mask? Okay. And that the pool, light switch, behind the toilet. Where else did I? Front door handle. And front door handle had the lowest number of bacteria. So what does it tell us if the shoe bottom, keyboard, water fountain, and cafeteria had high numbers of bacteria? Are there a lot of people going through there right now? Are there a lot of people using the cafeteria, using the water fountain, or the computer labs? No. So what does it mean if those places are dirtier? They're not dirtier because more people are touching them. Why are they dirtier? There we go. They're not getting cleaned. So shoe bottom represents the floor, right? So this is telling us that the floor, computer labs, water fountains, and cafeteria seats are not being cleaned. Right, because they currently have very low traffic. So it's not that they have a lot of bacteria because lots of people are touching it. It must be because they are not being cleaned. And then for the pool light switch behind the toilet and front door handle to be so clean, that tells us, right, those areas are getting touched and used a lot. Those are being cleaned regularly. Okay. All right. So we are now going to move to our last little chunk for today, 3.43 Bacteria Investigation Part 2. And so your do now question today was kind of prepping you for this. So in the background information here, it says, we now have a better idea of the types of environments and surfaces that are likely to have higher concentrations of bacteria. We also see that bacteria are pretty much everywhere, so it's not surprising that a young child like Addie came in contact with bacteria, especially playing so many sports. But how can we protect ourselves against bacteria? What can we do to kill them? Okay, so that's what you're going to brainstorm here in our next investigation brainstorm. So if you click on the blue link to the assignment, it'll open up the assignment and it looks like this. And the first question starts with what we discussed at the beginning of class. So what are some different ways we protect ourselves from bacteria? What are some different ways we can kill bacteria? All right, then it says number 2, we want to run one experiment that will show us how effective our predictions are. Describe in detail how we could set up and run an experiment to find out how well using hand sanitizer protects us from bacteria. What predictions do you make for your setup in number 2? And how will you know if the hand sanitizer was effective against the bacteria? So let's brainstorm this first one together here. I want to see how good hand sanitizer is at protecting us from bacteria, which means killing bacteria. How could I set this up? We know we have Petri dishes, agar, I have swabs, I can get bacteria, hand sanitizer. So how should I set this up? Okay, so Bryce said we can do a before and after. What does that mean? Be more specific. Who has an idea of what Bryce means when he says do a before and after? Who could help him?
Swapping the area before the hand sanitizer and then after the hand sanitizer.
All right, very good. So she said, okay, so I could swab my hand before I use hand sanitizer, put it in a Petri dish and see what happens. And then I could use hand sanitizer, swab again, and see what happens in the Petri dish. Okay, that's a good idea. What predictions would you make for that setup? What predictions would you make for that setup? What do we assume is going to happen? What are our predictions about what would happen in that setup? Right. There would be less bacteria after we use hand sanitizer. If the hand sanitizer is effective, what would we see or not see in our Petri dishes? The hand sanitizer is effective. What would we expect to see or not see in the Petri dishes? Right. We would expect to see no bacterial colonies growing in the Petri dish. So we've gone over the answers of 1 through 4. Okay. Your job is to read and answer 5 through 14. These are other experimental setups we're going to do. One is going to be with antibiotics. One is going to be comparing soap to hand sanitizer. And another one is to see how easily bacteria transfer off surfaces. So like if a surface is dirty, how easy is it really to get the bacteria off the surface onto our hands? Okay. So your job, again, you do have to answer 1 through 4, but we went over those together. Now you have to use your own brain to answer number 5 through 14. You have a little over 25 or 20 minutes remaining in class. Your next class starts at 9:50. So you have the rest of class to work on this. If you finish, submit it. If not, it's due tomorrow night. If you have questions, make sure to email me or drop them in the chat. Does anyone right now know of any questions that they have for me? All right, then you guys can start getting work. All right, so for those of us who are here,